Hi, I'm Dr. Mark Rothenberg, the senior author on the article entitled Vitamin D Receptor and STAT6 Interactome Governs Esophageal Epithelial Cell Barrier Responses to IL-13 Signaling that we have recently reported in the esteemed journal Gut. Thank you for this opportunity to allow me to present a audio video abstract. The basis for this study was conducted based on serendipitous findings that my lab had. We were examining the esophageal epithelial cell publicly available epigenetic data sets, and we were struck to find that the vitamin D receptor was particularly enriched in terms of binding sites and chip sequencing in esophageal epithelium. We were also struck by the finding that the IL-13 induced epigenetic changes, particularly the H3K27 effects, were associated with binding of vitamin D receptor throughout the genome. This was very statistically enriched phenomenon, as we reported in the paper. So we subsequently explored the mechanism whereby vitamin D may work in conjunction with IL-13. As you probably know, IL-13 is a critical factor in regulating allergic inflammation. One of its potent effects is to induce transcription, as well as pro-inflammatory effects and loss of barrier function in esophageal epithelium. And indeed, this is important because recently an anti-IL-13 antibody, dupilumab, has been approved by the FDA for the treatment of EoE because of its great efficacy. So in this study, we tested the effects of vitamin D on IL-13 responses in vitro, as well as in an in vivo model, and we finally translated this into human studies. As reported, when IL-13 is used to treat esophageal epithelial cells, one can see an expected change in the transcriptome, as well as EoE-like changes, including cellular proliferation, as measured by DNA stain, loss of barrier function, as measured by loss of filigrin, loss of differentiation markers, as measured by involucrin. Now, it turns out that the normal culture conditions for esophageal epithelial cells are low in vitamin D. When we normalize vitamin D with supplementation, we can see that the effects of IL-13 were dramatically inhibited. You can see loss of the proliferation marker, improvement in the expression of the filigrin gene, as well as the differentiation gene involucrin. We went on to examine this on a molecular level by showing in the same esophageal epithelial cells that the genome-wide changes induced by IL-13, as well as the epigenetic changes as measured by H3K27 acetylation chip sequencing peaks, were related. And this was controlled by the vitamin D levels. One can see that if you compare vitamin D low and normal levels under both readouts, that there are a series of genes that are unchanged and others that are changing in an antagonistic manner. In other words, if they were induced by IL-13, they'd be suppressed by the vitamin D, and vice versa. Other studies, as shown in the lower panel, revealed that there was co-localization of the vitamin D receptor and STAT6 at a very striking level. We went on to test this in vivo by examining IL-13 transgenic mice that have esophagitis with eosinophils and changes that are reminiscent of EoE that we see in humans. When the mice were on a low vitamin D diet, they had very dramatic changes in the esophagus. One can see here with the IL-13 induction, and this is an inducible transgene, you can see changes in the epithelial patterning and an infiltration of cells. Staining with collagen revealed increased collagen synthesis production, and eosinophils that are readily detected with the antibody against their granule protein, major basic protein. When we normalized the vitamin D, we saw an attenuation of these EOE-like changes, which you can see in the lower panel and quantitated in the right graphs. The eosinophil levels were decreased, the fibrosis scores were decreased by the vitamin D supplementation. We went on to further examine this in humans by showing that indeed in a cohort of EOE patients, vitamin D levels were relatively low, and the degree of eosinophilia in the esophagus, as well as changes in this pathology elements as measured by a histological scoring system, were both inversely proportional to the vitamin D levels. Taken together, the human data, with the human data and in vitro data, we have substantiated that under normal conditions, under homeostasis, the esophagus and the epithelial cells have a minimum response. However, in the low conditions, they're more susceptible to IL-13 triggering, and the vitamin D deficient state is seen in the EOE, and that this includes correlations with esophageal inflammation, eosinophil inflammation, basal cell hyperplasia, and other changes that we have now evidence that vitamin D may be playing a role in. So these findings really call attention to the interaction between the genome-wide effects of IL-13 and those induced by vitamin D. We're particularly calling out an antagonistic relationship, and these data indicate the potential therapeutic capacity of vitamin D agonists, vitamin D receptor agonists, in terms of treating EOE. This is particularly exciting since vitamin D receptor agonists are currently used for treating certain skin ailments, so that topical delivery may be amenable even in esophagus. Thank you very much for your attention, and I want to thank all the contributing researchers as well as the funding organizations which are acknowledged in this last slide. Also refer you to the websites, my lab, and also encourage you to go to Egypt Express, which has some of the data shared here today, and is a data sharing site which allows you to get access to many of the large data sets that we have published that can help you test your own hypotheses and advance this field and those of others. Thanks again for your attention.